Thank you very much, Alex. Dear colleagues, good afternoon. Claudio and the organizing committee, thank you very much for having me here. I'm waiting for the slides. Okay, here they are. So during the next 20 minutes, I'm going to talk about extracorporeal blood perfusion techniques for sepsis. These are my conflicts of interest. And the outline I'm going to follow, I'm going to talk first about pathophysiology and how does blood purification work. Then I'm going to list the different techniques that are available in 2018. And I'm going to try to say a few words about why we should continue research in this field. So as an introduction, I would like to remind you that when there is a bacterial insult, the body reacts with a double inflammatory response. With a pro-inflammatory component and an anti-inflammatory component at the same time. But the net balance is more on the pro-inflammatory side during the first days, and then after a few days, the net balance is on the anti-inflammatory side. And this immunoparalysis state that you have after a few days is extremely deleterious for the patient because at that moment, the patient cannot fight against secondary infections, cannot fight against viral reactivations such as CMV or HSV. And that's why actually when the majority of the patients who die from septic shock, they die at that moment. The other thing I would like to say as an introduction is, of course, it was already said, we have a new definition of sepsis, but the first sentence is very important. Sepsis should be defined as a life-threatening organ dysfunction caused by a dysregulated host response to infection. What does it mean? It means that if we work in the ICU, if we are intensivist, then we need to have a little bit of knowledge about immunology, because now in the near future, I think immunology will be part of our work. And how we have a dysregulated host response. So how can we get back to a kind of immune homeostasis? Well, we can do it thanks to different strategy. We can give some molecules, like some immunostimulants. Some molecules are under investigation at this time, IL-7, interferon gamma, GM-CSF. So this is a possibility. And the other possibility is to give to our patient some of these techniques called extracorporeal blood perfusion therapies, which are also at this time under investigation. So how do these techniques work? Well, we don't know. We have no idea. We have only hypotheses. Because first of all, we should think which target we should be focused on. Should we try to remove endotoxins? Should we try to remove cytokines, inflammatory mediators, such as TNF, IL-4, IL-10, IL-6? Should we try to modulate the gradient of cytokines, chemokines between the tissue and the circulation? This is a hypothesis from Professor Kellum. And when I'm trying to explain this one here, you have the circulation here and the tissue. Let's say it's lung. So you have pneumonia, and you have three players. You have cytokines, you have the bacteria, and you have the leukocytes. So if you run the blood through a blood purification device, whatever the one you choose, then you will be able to increase the gradient of cytokines between the circulation and the tissue. And this gradient is very important because we know that this gradient drives the leukocytes to the site of the infection. It tells the cells, okay, you have to go to the lung because this is where the infection is. And so the bacteria can go to the lung and kills the bacteria. So of course, this is also an hypothesis and needs further work. We have also other hypotheses. When you put together a sorbent and a leukocyte, then they're going to interact in some ways, and you can have some modification of the expression of some markers at the surface of the leukocyte. For example, HLA-DR is very important. these devices, they can also absorb the leukocytes by themselves. So you can also decrease the number of leukocytes in the blood thanks to this device. So we don't really know which strategy we should follow, but you can see that there are different hypotheses. And here, talking about the absorption of leukocytes, you can see on these pictures that you have some devices. This is divinylbenzene beads. So they are able to absorb the leukocytes directly at the surface of the device. So what are the techniques available in 2018? We have now a large panel of techniques: high volume hemofiltration, cascade hemofiltration, plasma exchanges, coupled plasma filtration adsorption, hemoperfusion, and all these new membranes recently developed, high adsorptive hemofiltration and high cutoff membranes. I'm going to say just a few words about all these therapies. What about high volume hemofiltration? It was a technique studied a lot in the 1990s and in the early 2000 years, and then came out the study, the IVOIR study from Olivier Johannes-Boyau and Patrick Honoré. They planned to enroll 460 patients and to randomize them in two groups, 35 ml per kilo per hour versus 70 ml per kilo per hour. And they wanted to look at the survival. The problem is that they could enroll only 140 patients. And I think this is important. The study was completely negative. And now, because of this study, the number of teams across the world who use high volume hemofiltration has drastically decreased. What about plasma exchanges? We have an interesting paper which was published a long time ago in a good journal, showing an interesting signal of plasma exchanging in patients with septic shock. It was a study from a Russian team showing, again, survival improvement when the patients received plasmapheresis. And the problem, this study did not have a huge impact in the medical community, probably because the randomization did not do a great job, because the patients from the control group were a little bit older, it was statistically significant, and the sites of infection also was a little bit different. But I'm a little bit surprised that this work was not followed by other works looking at plasmapheresis in sepsis. Coupled plasma filtration adsorption, it's an Italian technique, so we have to talk about it here. It's a hybrid technique, so this is interesting. The blood goes first into a plasma filter, and then the plasma goes through a resin, which is able to absorb a lot of cytokines. So the cytokines are absorbed on the resin, and then the plasma, after it has been cleaned from the cytokines, is going back to the circulation, to the circuit. And then the second part of the circuit is hemofiltration. And so you can do renal support. So it's a hybrid technique. I mean by that you can do blood purification for sepsis during in this part of the circuit, and renal support in the second part of the circuit. So what data do we have with CPFA? We have a large randomized control trial called COMPACT-1. This was a multi-center randomized control trial done in Italy. Standard treatment versus standard treatment plus 5 days of CPFA, 300 patients from 18 centers. They enrolled 200 patients only because the study was stopped earlier because there was absolutely no signal, no difference in terms of survival. But interestingly, there was a sub-analysis showing that if you give more CPFA to your patient, then there was a trend to survive better. So there was a second study which was performed also in Italy, the COMPACT-2 trial, which was supposed to assess a higher dose of CPFA in patients with septic shock. And the bad news actually is that a few days or weeks ago, I've learned that the study was stopped because of bad results in terms of survival in the patients who received CPFA. So at this time, the study is stopped. It will be a negative trial. I hope it will be published because we need further understanding why this study was stopped like this. So hemoperfusion, I'm not going to talk about it because Claudio is going to do it just after me. So I'm just going to focus now on these new membranes. So high adsorptive hemofiltration, this is the membrane called Oxaris. This membrane is a renal replacement therapy membrane proposed for CRT. And the target of this membrane is double. You can target the endotoxins and the cytokines. So it's interesting by this capacity of adsorption. So why is this? It's because this is an N69-based membrane with polyacrylonitrile. And because of this, the membrane is able to absorb the cytokines in the bulk of this membrane. IL-4, IL-10, TNF are absorbed at the bulk of the membrane. And in addition to this, the company has added a positive charge at the surface of the membrane, polyethylenamine, which is able to absorb endotoxins at the surface of the membrane. So again, you can absorb cytokines in the bulk of the membrane and endotoxins at the surface of the membrane. This is an heparin coating membrane also in order to compensate for the increased thrombogenicity due to this positive charge at the surface of the membrane. So by adding heparin at the surface of the membrane, there is no anticoagulation problem. We have very limited data with Oxaris. We have animal works, we have in vitro works, and I'm just showing here the latest data done in the lab. It's a study conducted by Professor Callum. It's a study comparing 3 different blood perfusion devices, the Oxaris membrane, polymyxin B, Hemoperfusion, and the CytoSorb technology. It's a study, an experimental work, it's an experimental work with a closed circuit, and the investigators run the blood in the circuit for 2 hours after they spiked the circuit with different quantities of endotoxins and cytokines. And then at different time points, they looked at the capacity of the devices to absorb the inflammatory mediators. So in terms of endotoxin absorption, there was no difference between the polymyxin B hemoperfusion device and the Oxaris filter. CytoSorb was not able to absorb LPS, but this is not the target of this device. So it was not surprising data. In terms of absorption of inflammatory mediators, there was no difference between Oxaris and the CytoSorb technology. They were both able to absorb a lot of different mediators, actually almost 30 different inflammatory mediators in that study. So I believe the 3 advantages of this Oxaris membrane is, first of all, this is also a hybrid technique. You can do both at the same time, kidney support and blood transfusion for sepsis. And also, it can target the endotoxins and it can target the cytokines. We don't have a lot of human data, but they are coming. If you look at clinicaltrial.gov, you have several works coming that will be published, I think, in the next 2 or 3 years. And now I'm going to talk a little bit about high cutoff membranes. So the idea is to increase the size of the pores of the membrane in order to increase the spectrum of inflammatory mediators you want to remove. The problem with these membranes is the potential removal of albumin, and you don't want to remove albumin in your septic patients. So the compromise is to use these high cutoff membranes in a diffusive mode, not in a convective mode. You are not going to force the passage of albumin in the effluent. So use them in a diffusive mode and with a reasonable cutoff. Of course, if you use a membrane with a cutoff of 150 kilodalton, you're going to lose some albumin. But if you use a membrane with a cutoff for 40 or 50 kilodalton, the so-called super high flux membrane, then you will be able to remove inflammatory mediators, but not the albumin, because the molecular weight of albumin is 68 kilodalton. So this super high flux membrane has a cutoff between 40 to 60 kilodalton. And the idea, this is the MIC2 membrane here, the green line. You increase a little bit the cutoff, and then you get more inflammatory mediators from the blood without too much albumin removed. That was actually showed by the team of Detlef Kinge-Miles in Germany. They looked at, I think it was 15 patients, and they run super high flux continuous hemodialysis, and then they found that the solute clearances was quite high. Not only for small molecules, urea and creatinine, but also for middle molecules. Like you have markers here of middle molecules, myoglobin, free light chain kappa, free light chain lambda. And when you look at the clearances, you can see that they are quite high. And again, look at the clearances of albumin, very low. You look at the y-axis, 0.02, 0.03 ml per minute. So this is very low clearance. In that study, the investigators didn't have to infuse additional albumin to these patients. So to finish, during the next 5 minutes, I would like to explain why I believe we should continue research in this field. The problem is now when we go to meetings, we face some people, that they are telling us, okay, we have different large randomized controlled trials that are negative with blood purification techniques. So we should stop talking about that. And I strongly disagree with this, and I'm going to try to explain why. I think this is totally wrong for 6 reasons. The first one is this is wrong to say we have only negative trials. We have negative trials. I'm going to display them a little bit later, but we have also positive works. This is a positive meta-analysis from, I think, Professor Cullum's group. This is the study from Professor Ronco, published in JAMA, the UFA study. It was positive work in terms of hemodynamics results, in terms of respiratory parameters, in terms of SOFA score. There was even a signal on survival. Positive results also in sub-analysis of the COMPACT-1 study. Positive study also with another meta-analysis with CPFA. Positive sub-analysis again, which for a study which is not published yet, the Euphrates study, in a subgroup of patients, we have also a signal in terms of improved survival with polymyxin B hemoperfusion. Second, these studies are the ones who are negative, the large trials, the IVOIR study for high volume hemofiltration, Abdomic study for polymyxin B hemoperfusion, and COMPACT-1 for CPFA. Well, these studies also have a lot of limits. And for example, the IVOIR study, they were performed by good friends, but they wanted to enroll 460 patients, as I said earlier. They enrolled only 150 patients. So when you do this, you cannot draw very strong conclusions. Well, if you don't believe me, then you can ask the sepsis experts what do they think about blood transfusion techniques. Well, the question was asked to the sepsis experts from the sepsis campaign, and they could not make any recommendation. We make no recommendation regarding the use of blood transfusion techniques because the evidence is too low in favor or against these therapies. So that means we should continue research. We have studies, we have techniques that were deeply assessed, but also we have other techniques that need additional work. And this is, I think, plasma exchange, as I said, hemoperfusion outside polymyxin B, all these new filters, very limited data. I think this is also a very important slide. When you test a molecule or a therapy or a device, if you take a too large population, then you may miss the signal on the subgroup of patients, which is interesting. So let's imagine one of the blood perfusion device has a positive effect on a subgroup of patients here. It could be neutral on another subgroup of patients, and it could be deleterious in another subgroup of patients. But then this positive effect, you are not going to see it if you look at the entire population. So that's why it's very important to select very precisely your target population in these trials. Six, finally, we had at the beginning of the Congress a nice conference about technological progress in our field. This is true. We face new technological progress every day, but I think technology is going faster than our medical knowledge. And so we have now many unanswered questions. We don't know really how to use these devices. I tried to explain you this. We don't know which population we should target. How do these therapies exactly work? We have hypotheses, but we need to address them. What to ultimately remove? Should we remove endotoxins? Should we remove cytokines? Should we remove the activated leukocytes on the sorbents? Even should we remove bacteria directly? Because now we have the possibility to remove bacteria. This is a work from Harvard in Boston. They are able to remove directly from the blood the bacteria. Also, this is the same for the viruses. On the other side of the United States, in San Diego, there is a device which is able to remove directly from the blood some viruses. So what to remove? Endotoxin, cytokines, leukocytes, bacterial viruses. This is an unanswered question at this time. Conclusion, I would say that sepsis, it's frequent, poor prognosis, very expensive. In 2017-18 right now, immunology is now part of the sepsis definition. The patients, they die of septic shock because of immunosuppression. All these techniques, all these extracorporeal blood perfusion therapies, they can be considered as a potential adjuvant treatment for sepsis. We have some promising data, and the new membranes, I believe they are quite promising. We have negative trials, large trials, multi-center, but were they well designed? Did they target the right population? So now in 2018, we have many unanswered questions. Which patients, which timing, which technique is best? How do they work? So that's why I believe we should continue research. Thank you very much.